I think it's a great initiative that Manju has started. It will be very helpful for the students because when you are early in your path, it's nice to learn from others' journey. How they have done things and what they have learned. So, um, congratulations, and I'm so happy that you guys are doing it.
Yeah, thank you, Rupali. So my slide is there. I mean, you can.
Yes, ma'am, it's visible.
So I
Manju
think we
is
can start the program for today.
Ma'am, the slide is not visible. Your screen is visible, but you will
Yeah,
have to share
yeah. I will ask Professor Ramakrishna to have some opening remarks, and then we can proceed. Yes, sir.
Okay, I will start now.
Yes, sir. Sure.
Okay, so good evening, everyone who are attending from India. So we have today Dr. Manju started this series of the possibilities for a science enthusiast. So this is the second talk in this. So today we have Dr. Rupali Gandhi who is medical director at Sanofi Genzyme from USA, and she will be talking on success is a journey, not destination, from academia to healthcare. So Dr. Manju will introduce the speaker. I want to say a few things here related to the topic. Success is journey, not destination. So this is something I keep thinking a lot of times during my entire career. So something related to that is we all have some goal in life, right? We want to become this or we want to achieve this. So we are always running after that particular goal. So in that goal post, what we have, we generally don't enjoy that journey. So it's important really to enjoy that journey and get maybe some little small successes we have during the journey that we need to enjoy. I think that is the important take home message is to be happy with what you are doing, because in our journey for that final goal there is so much stress, okay, that we get so many negative things happening. In the negative things and stress, we also have situations where we find small successes and we face happiness. So we have to enjoy that, and once we get to one goal, then we may have find a second goal or third goal. So all this experience we are having, we have to learn from there, and especially for students, this is important. Learn from your experience, whether it is a positive or negative, both ways we will learn and enjoy that journey. I think that is what we are going to go today. And Dr. Rupali, I was seeing at her profile, she did some excellent work during her academic career, PhD, postdoc, and she was assistant professor. So then she changed her field from being from academics to that and the industry, and a little bit away, more from the management viewpoint. So we will listen from her what was her journey and how to enjoy that success during the journey. So with this, I will hand over to Dr. Manju to continue.
Yeah, thank you, sir. And I will say words of thanks for Professor Ramakrishna, as well, as he always encourages such programs. And if as long as you are logistic with him, you are working on a logical idea and can explain things, you can't expect to hear a no from him. So he's always there for us. So thank you, sir. Yeah. Let me go to the. So all of you can see the slide.
Yes, ma'am.
Yes, ma'am.
So on this note, I formally welcome our today's second speaker of the series, The Possibilities for a Science Enthusiast. We have with us Dr. Rupali Gandhi, and I would like to take this privilege to introduce her with a top to bottom approach, so that students can relate with every bit and piece of this event, uh, you know, uh, in a deeper sense. So Dr. Rupali did her BSc Microbiology from Delhi University. She pursued her master's in fermentation and microbial technology from Guru Nanak Dev University from Punjab itself. And there onwards, she went ahead with her PhD with the major area of work in the field of immunology at the National Institute of Immunology in affiliation with Jamia Millia Islamia. And it is from this time that I got to know her and is sort of associated with her. During her PhD, she worked on the autoantibodies in context of systemic lupus erythematosus, with excellent work profile and the project and the way I've seen her working. She went ahead with her postdoc at Brigham and Women's Hospital, Harvard Medical School, and working in the area of autoimmune diseases, specifically the neuroimmune diseases, I would say. And from there onwards, she has worked on different positions as a instructor to many more science scholars in an international setting. And alongside, she had experience on the managerial part of a laboratory. So with these all rigorous academic research and management based experiences, she was all prepared with the gusto to go ahead for an assistant professor position at Harvard Medical School at Brigham and Women's Hospital, Department of Neurology. She has shown her scientific expertise in the field of immunology, autoimmune diseases, and neurological diseases. And with this kind of academic profile and research output, which any one of us, would love to have, she made a major move towards moving into a healthcare-based industry system. And at present, she is working as a medical director at Sanofi Genzyme USA. So with this, I would just, knowing these details, I went back to the Sanofi Genzyme site, and this is what I saw. So I thought I would just share it with the students with the perspective that the Sanofi Genzymes, the motto is central to patients and science, at the center of everything they are doing. So that itself speaks a lot. And the rest we have with us Dr. Rupali Gandhi, who will acquaint us and make us familiar with what all that goes on behind such a successful journey. The journey part has to be emphasized here, and to reach the pinnacle of scientific excellence, I would say. Yeah. So Rupali, the stage is all yours. Thank you.
Thank you, Manju. Thank you, Manju, so much. And I am so proud of you, for initiating this particular series. And I think the students will get really benefited because I have learned a lot from others' experience and my mentors. And I think this is a wonderful program. So thank you for organizing it and inviting me. And thank you, Dr. Ramakrishnan, for your support for this program. And thank you to the whole department, I'm sure, must be involved in such initiative. I want to start with thanking all the students, for joining me on a Saturday evening. I'm sure your lab experiments must be running. There are a lot of family needs that you might have to take care of. But thank you for joining me today to learn about my journey. And I hope, this particular talk could help you in carving your own future. So here I will share my slides, and I am actually, after my talk, I'm actually looking forward to a lot of discussion with you guys, answering any questions you guys have. So here goes my slide, and Let's see. Can you guys see the slide?
Hello.
Can you guys see my slides?
Yes, Dr. Gandhi, it's visible.
Awesome. So as Dr. Ramakrishnan said, success is a journey, not a destination. And this is what I want to highlight with you,
Go on,
like,
right? Yeah, it's in the sense we need not worry about more if more people join then.
Dr.
Manju, you are
Manju Jain,
another
can you mute your microphone, please?
Okay. I think Manju was on a call. So success is a journey, not a destination. And I think we have to remember it's an important point. So you guys must have, your next step sorted out. What is the next step? But I would say, that next step is not all your life. So as you go to your next step, you will find that your interest and your passions might change, or even you might think that, okay, maybe I want to try a new career, I want to learn about a new thing. And career is a vast, there are so many opportunities available for all of us, but sometimes we don't think about all those possibilities available to us. So hopefully during this talk, I will reflect on my journey, and you could maybe learn some pieces from that. So in this particular talk, I'm going to talk about in three different sections. So first section, I'm telling you, what has inspired me to be who I am. In second section, I want to share some of my academic research that I did, and I will promise you that I'll keep it high level. And if I don't keep it high level, please ask me a lot of questions after the talk. And in the third sections, I want to bring to you some of the learnings that I had during my career. So many years ago, I was born in this beautiful India, and I'm very proud to be born there. And I know India is at a very different level and a very different mindset right now. But in my childhood, the main trouble for girls born in India was they were like a burden for the society. They were burden for family. And in my family, we were three sisters. And when my third sister was born, my whole family was like kind of in a shock, or in a kind of sadness that that, oh, there is another girl. And there were a lot of talks in my family for exchanging my sister for another boy or something. And that made me think, why girls are not appreciated, why girls can't do what boys can do. So and it still resonates with me a lot. I feel very hurt by that concept during that time, and I'm sure India is at a very different level at that time. And I think one thing that I learned, that why girls was considered as a burden, because of all the effort parents have to put for the girls wedding, that one night wedding day, how much money parents have to put. And then they let the girl go into another family, then she's not part of your family, she's another family. And I have very different views about that, and I feel like girls can do as much as boys can do. And I know now society is at a different level, and there are so many programs encouraging women. But I am wishing and I'm hoping that you guys are at a stage, where the girls are as much respected, and their talents are appreciated as much as boys. And I'm sure the society is at a different level. But this is what has inspired me to be something, because I did not want to be a burden on my family or burden on my husband. I wanted to see that girls with their hard work and with their passion can do as much as anybody else can do. So I want to thank my parents. When those times, it was not easy for my parents to think out of the box. They have let me do my PhD, and so many family members. I know my grandparents were very upset with my mom and my dad that they are not marrying me, early in my life, and that, all the marriage availabilities will be shut down once I'll finish my PhD. But, my parents believed in me. They gave me the opportunity to study and to be who I am, and also to my sisters. So I'm very thankful and being very lucky to have them in my life. As Manju said, I did my PhD from National Institute of Immunology with an amazing mentor. And I think who I am today is because, what he taught me about science, about being honest, about being transparent, and how to do science in an honest way, and how to think about patients. So, I did my PhD studying B cell biology in lupus patient, and my only aim was, what is wrong with these autoimmune diseases. Maybe understanding of that could help us to really find treatments for them. And I am very sad to hear, there are so much research is going on, but still in a lot of neurologic diseases and autoimmune diseases, we still don't have a good target. But, we have a lot more understanding, how things were like 20 years back when I did my PhD. We are in a very different science era. And in my next projects, I think I'm coming back to lupus biology. So I think it's very interesting to understand these autoimmune disease and find new targets from that understanding that could really help patients. And here in this slide, I'm showing one way, because, after I did my PhD, my only thought was I have to go for postdoc fellowship in US in a good college, because that's what most of the PhD students from my institutes were doing. And I think, and Manju knows that experience as well. A lot of people who study research in at National Institute of Immunology are encouraged to go for a postdoc fellowship. And I think that one path actually opens up many different paths for us. But at that time, the only thing I knew that I have to do a postdoc fellow. So I came to Harvard Medical School to work on another autoimmune disease, but which was also neurologic disease, which is called multiple sclerosis. And I'll talk, in the next few slides about what that disease is and what I have done in that particular disease. So, it was not easy coming from India at that time, being a very pampered girl and coming to Harvard Medical School in Boston. Now we are very much connected by Skype and WhatsApp. At that time, we didn't have that many options to connect with your family. So it was a hard decision. And to be honest with you, it was not easy. It took me, I think, almost 3-4 years to really make Boston as my home, and felt like, okay, I belong here now. So, when you will go to your next level, be prepared for that. It's not going to be easy, but I'm sure it will be worth it. And in next few slides, I want to share with you some of the research that I've done during my academic career. And I'm only picking up 3 projects, not to bore you on a Saturday evening. And I'll try to keep it very high level. So during my academic career, I worked on, I think, maybe 15 to 20 different types of projects. I had about 45 different publications, peer-reviewed publications, presented at various national and international conferences. I was a reviewer for multiple manuscript or journals, and also reviewer for some of the grants for MS Society and for NIH. So the first topic I want to talk to you about, I worked during early career in academia, was to understand the immune system biology. So I did some work on T cells, regulatory T cells, dendritic cells, monocytes from multiple sclerosis patients, and understand how these cells are different in multiple sclerosis patient compared to healthy controls. To my surprise, I found that all these markers, the main trouble in all these autoimmune diseases is we don't have black and white signature. If we understand what particular cell goes wrong in autoimmune or neurologic disease, we might have an easy target to address. But I think there is, in these autoimmune and neurologic disease, there is spectrum of things that goes wrong. And I think that's where we want to take science, is to understand that particular spectrum, and that spectrum of what is altered in the disease versus health is very diverse. It can be very dependent upon a patient, and that's what I think right now we are working on, is the personalized treatment. So in this particular project, I worked on regulatory T cells and understanding what induce these T cells. So I worked for inducing the regulatory T cells, and this was mostly on all human T cells. This work was inspired by one of my colleague, Francisco Quintana, who has done a lot of work in the mice biology, and we collaborated to understand the importance of this pathway in inducing regulatory T cells. So AHR is a receptor that is present on liver, kidney, and lungs, and also a lot of immune cells. And this pathway is mostly involved with taking care of toxins in the body. But we found out that if you activate this particular pathway, we can induce regulatory T cells. And I keep saying the regulatory T cell. What are regulatory T cells? So regulatory T cells are kind of what I say is like cops for your immune system. And I do not know where you are in terms of your understanding of immunology. Many of you already might know about these cells, and some of you might not have heard about it. But these particular cells are like cops, and they keep your immune system in control. So as you know, whenever there is a virus or a bacteria attack,
so Rupali, you may please, sorry to interrupt, you can click the stop, the hide thing, so that we see the details of your slides.
Oh, you couldn't see? Sorry, you were
No,
not
no,
able.
it was just a little thing at the bottom, but then yeah.
Oh, okay, sure.
Please.
So awesome. Everything else is good, Manju? Yeah. Okay. So these regulatory T cells are like cops. They keep the immune system in check. So whenever there is a pathogenic attack or a infection comes to your body, our immune system mounts an immune response. And so these regulatory T cells, once the infection is gone, they tell the immune system, all is good, now calm down. We don't have to mediate an inflammatory response. So that's why you see here a balance that shows fire, which is, when immune system is active and mounting an immune response, and that these anti-inflammatory responses by regulatory T cells keep the balance in control for our immune system. So here, this is a lot of detail, lot of data here, but I don't want to go into too much detail. But I wanted to tell you that when we activated this particular AHR pathway on T cells, we generated TR1-like of immune cells. So these cells were the ones that were functioning through IL-10, which is one of the immunosuppressive cytokine, and also by engulfing the inflammatory cells or the dying cells. And when we activated this particular pathway in presence of another cytokine, we found that these particular cells become into FOXP3 cells, which are more like natural regulatory T cells, which are more commonly present in our immune system. And maybe, you guys might have heard about the FOXP3 regulatory T cells, which are very popular cells in human as well as mice biology. So this particular project, I don't want to go into too much detail, but what I said in summary is that, when you activate this particular pathway one way, it induced one type of regulatory cells. But if you add on some other cytokine mix, these cells become another kind of regulatory cells, and they function in a different way. And we also try to understand what are the upstream molecular pathway that was involved in generation of these cells. And I'm very thankful that actually this particular study, we published a lot of papers. I think I was involved in 2-3 of them in the human biology, but my colleague had continued this work in mice biology. And I know they are now testing, if we can induce some of these cells to really help in the autoimmune diseases such as diabetes and other autoimmune disease, to generate some of these cells in vitro and then take them to treat patients who are suffering with some of the autoimmune diseases. Okay. The next project, will need some understanding about what multiple sclerosis is. So multiple sclerosis is an autoimmune neurologic disease. So what happens, the multiple sclerosis means there are lesions in patient's brain. That's why the name comes as multiple sclerosis. And this particular disease, have different trajectories. Patients have different types of disease experience. Some of them have relapsing remitting course, which is shown in the blue line here. Patients have disease onset and then patients get better. As you see the arrows going up, the line going up means the patient are having the disease and then they get better. In the primary progressive, which only present in 10% of the patient, these patients start to get worse right from the beginning. And the patients have the disease and then they continue to get worse. And we don't have, I think we have now one treatment in the market for last 3-4 years, but there was no treatment for this disease for many, many years. Going back to relapsing remitting patients, most of the patient, about 90% of the patients, start the disease as a relapsing remitting course. And when 15 years back, when I started to work on multiple sclerosis, there were only 4-5 treatments available. But today we have more than 20 treatments available for multiple sclerosis. But most of them address the relapses of the disease and not the progressive part of the disease. So now the biology is to understand what causes the disease progression and how to inhibit that progression or take care of that progression. So about 50% of the relapsing remitting patient becomes secondary progressive. So these green dotted line, as you can see here, shows that these patients have relapses during early on in the disease course, but then they become worse on a continuous basis, and that's why these patients then are called progressive patient, and this disease state is secondary progressive disease. So this particular study that I worked on took, I think I was working on that particular study for 7-8 years, and I think this was very important for me because I was thinking, if we understand the biomarkers for these neurologic diseases, that can help us classifying the patient in a better way, so we can treat these patients better. Going back to the personalized treatment, so understanding the patient more based upon the biomarkers, so we can find the right treatment for the patient. And some of the uses of biomarkers are, to early diagnose the patient, because if you catch the disease early on, you treat the disease early on, you can find, maybe you can treat the patient better. A prediction of the disease outcome, if I know that a particular patient is going to progress later on in the disease course, maybe I will choose a different treatment for these patients early on so that the patient could benefit and can live a better life. These biomarkers also help us to measure the disease activity. If I know the patient is going through relapse, and if I can treat the patient, maybe patient's disease progression will come later on, and maybe patients might not have a disease progression later on. And also, to understand, we need the treatment outcomes, right? If I have a patient being treated on one particular drug, but for 6 months I am not able to figure out whether the patient is getting benefited by the treatment or not, then it's very hard for the patient, because for 6 months the patient is just suffering. So if I have a biomarker that can help me tell that patient is actually benefiting from the treatment, then I can make better choices for the patient early on. So that's what I think the biomarker research was very important for me, to understand the patient biology so we can treat the patient in a better way to help patients live a better life. So our objective was to find different biomarkers to identify the disease subtypes, predict the disease worsening, and to identify the treatment response. So the biomarker that I focused, one of the biomarker, I worked on several biomarkers along with my colleagues who had different expertise, but one biomarker that was very hot in 2006, 2010 was microRNA. So microRNAs are small RNA molecules that actually binds to the RNA molecule and tells this RNA molecule to degrade. And in 2006-7, there were several studies that showed the importance of these microRNAs as biomarkers, and they can be measured in serum and plasma from the patient. So we looked at this microRNA biology to understand the disease, MS, multiple sclerosis specific biomarkers for patients. So this particular slide shows that biomarker study is not one project that you do. So it is like a commitment, like a lifelong study. So it's just like a clinical trial, where initially you do some discoveries, like how we do discoveries in the lab. Then you have to confirm those discoveries at multiple level, and that's what is shown here as validation 1 and validation 2. And then once you validate in a significant number of patients, you have to develop the assay so it could be used in any patients all over the world, and in a very fast way and in a very easy way, right? And if you think about these biomarkers, we know that there are biomarkers for diabetes, and how this insulin test is so helpful for patients to know about the diabetes, right? But for a lot of autoimmune and neurologic disease, we lack these biomarkers, and we need to spend a lot of resources, time, and have a lot of patients to really develop, significant biomarkers in these areas as well. So this was the different steps that I took for my microRNA biomarker study, and some of them I'm showing here. Maybe we can have a quiz at the end of the talk, and I'll quiz, what are the biomarkers that we found out in microRNA based biomarker for different disease. And this particular slide shows how we found biomarkers which could differentiate a multiple sclerosis patient from a healthy control, which could differentiate a progressive patient from a relapsing patient. And we also found some biomarker that were related to the disease activity, which was EDSS is one of the measure for multiple sclerosis patient, and we found several microRNA biomarker which could relate with the disease in MS patients. And here is the validation study. So once we discovered the biomarker, we confirmed it in about 100 patients in our cohort. Then we also collaborated with multiple cohorts. There were one cohort in USA, one cohort in Lebanon, and one cohort in Sweden, and tested these biomarkers again so that we can confirm that these biomarkers were not specific for Brigham and Wound patient. Worldwide, wherever we go, we can check some of these biomarkers and could check in different types of patients and from different ethnicity and different geographies, and can confirm that these biomarkers are good tool to develop further for the clinical testing. So this particular study led to many different studies. We tested biomarkers in many other diseases beside multiple sclerosis. We checked biomarkers in ALS patients. We also checked biomarker in other neurologic disease, and some of that work was actually not published as well because of my decision to move from academia to industry. We found that these biomarkers are related to some of the MRI outcomes in neurologic disease. We found how microRNAs in the stool could help to also differentiate a multiple sclerosis patient from a healthy control or other neurologic disease patients. And we also found these microRNA, how to develop them as a more detailed biomarker. I worked with 5-6 different institutes across USA. They were also part of the same funding that I had from NIH to really understand this microRNA biology, and then to define the test that we could use to identify these microRNAs in serum and plasma. So that was a very, very exciting project for me. And I'm actually looking back, I'm very proud of these particular studies and the collaboration I had with other colleagues for this. One other study, and I think was very novel, that I loved, was understanding the gut microbiome. That idea of working with the stool samples disgusted me when I started this particular study. But then, I understand, gut microbiome is actually very, very important in understanding various diseases, because, we all are inhabitant with millions and trillions of bacteria in our gut, and that bacteria is actually help us digest our food and live a healthy life. And if there are changes in that gut microbiome that could lead to diseases or unhealthy states. And now, when I started in 2010, there were not a lot of studies to work looking at the gut microbiome, but now we understand various assays that we could use to understand this gut microbiome. And there are several studies going on in various diseases to understand gut microbiome and how they are changed in disease compared to health. So this particular study involved about 100 patients, and this diagram shows what we did. Besides testing the gut microbiome, we also tried to do some serum analysis and some cellular analysis, just to show that if this gut microbiome is changing, it's actually affecting the immune system. It was changing the immune system. And here is a slide that shows the data, and lot of colors, lot of information. You don't have to remember it, but just wanted to show, like, kind of give you a feeling that when we tested MS patient, multiple sclerosis patient who were treated or not treated, or MS patients, multiple sclerosis patient compared to control, there were different phyla and different genera of bacteria present in their gut compared to healthy controls, or compared treated patients to an untreated patient. So these graphs actually shows what different bacteria or their genera or their phylum were changing in disease compared to healthy controls. And we tested it using 2 different technologies. At that time, we were not sure which one of them is better, because that was very early on in, in the microbiota science. So we tested during 2 different tests, and we got similar results, which encouraged us to show that, whatever we were doing, we were on the right path. It was not a false signature. And this particular study, we looked at the microRNA also in the stool samples to confirm, what we found in the serum in the previous project that I was talking about. And then also one of my colleagues did a probiotic study, where she fed the probiotic, which we know are the good bacteria, to multiple sclerosis patient, and see, how that if that can help the patient feel better. And that was a very small group of patients, but it seems like there was some benefit of treating the patients with probiotic in multiple sclerosis, that was supported by the study. Beside these human studies, I know there were a lot of mouse studies that my mentor continued to do, taking the bacteria and putting them in mice and seeing, if mice disease get worse or better, treating with that particular bacteria. So it was very exciting project, and, very innovative and novel at that time. And I loved working on these projects. So, once from spending like 13, 14 years in academia, doing multiple different research and working, like I said, working on multiple different projects, I wanted to do something different. As you grow, like I said, your passion change, your interest change. And my whole, I understood, like in 2017, and I was always feeling I want to work for patients, and that's why I was doing the projects, I was doing research that can benefit the patient in some way. And as I was doing those research projects, I was collaborating with a lot of industry here in Boston, and I found out that a lot of my research interests are actually matching with what industry is trying to do. So I thought that why don't I try and make this jump from academia to industry to explore how can I help patients in a different way. It was not an easy decision because I spent all my career to be an assistant professor, to be a professor at a university, and I was very doubtful of my that decision, that am I making the right step? Is this the right step? Because 20 years I've done one kind of thing, and now changing my path, will I, is it going to take me in the right direction? But I think one thing that I kept in mind, I want to work for patients. I want to build science that can help patients, that can cure diseases. And that passion actually took me the right path. And now, being the medical director, which is part of the medical affair at Sanofi Genzyme, I my major role is to provide information to physicians so they can make the right choice for the patient. And that actually fulfills me a lot, thinking that I'm working for patients, I'm working for science and doing the right thing for the patient and for the doctors. So being part of medical affair, I learned that how PhDs could work in a very different way, coming different from lab work. So I always thought that being a PhD you can only do research, but I found out being PhD you can do so many different things in industry. And I collaborate, being part of the medical affair, with medical research, where we look for what are the additional data available for the patient, and how can we make our treatments better and more affordable for the patients. We work with medical science liaisons who actually deliver all the safety, all the efficacy outcomes from our products to the doctors, so they can make the right treatment choice for the patients. We work with medical education, we work with medical communication, who help to train the team, both commercial team, the marketing team, which actually goes out and sell the treatment or the drugs, and also the medical team who actually goes out and talk to HCPs about the treatment, goods and bads. So I think PhDs can, like for last 4 or 5 years, I learned that PhDs can do so many different things just beside doing the research work. And I'm happy, I'm sure there will be a lot of questions from this particular my role, but I'm happy to address them after my talk. So this is my journey so far. I did my PhD in 1998, joined the postdoc fellowship in 2004, became the instructor in 2008, where my role was not very much different, but, it was more permanent position in the academic career. Became the head of the biomarker around 2010, was managing the biorepository for 2,000 MS patients, collecting their serum, plasma, stool samples, TS samples, and everything from these patients, so we can find the right biomarker and also was leading a team of wonderful collaborators or colleagues who have expertise in identifying different types of biomarkers. Became assistant professor in 2013, and I think that was the day when I thought the dream come true, because all my life I thought that, okay, this is what I want to be, and this is what I want to do. And I loved my career, 14, 15 years staying in academia and doing wonderful project. Taking a water break. And then I joined associate director in medical affairs in 2017 and became medical director in 2020. I was in the US medical affairs right now, and then now I am looking to go into the global medical affairs team, which I hope will work out in next few weeks. So just wanted to tell from this particular slide, one thing is, whenever we go to our next level, it's not always a promotion. It might not always be, increased salary, increased responsibility, or increased managerial powers. It can be a parallel move. It can be something that, maybe it is opening up for you to learn a different scope or different types of work. So just from this path, I learned that, promotion is, or, taking next step is always not a promotion. It can be a parallel move, but it can be that it can open up a new learning or a new understanding or a new passion. Going to the next slide. Here in this slide, I show that, once you do your master's, once you do your PhD, don't think that you have only one track, which I thought, that I had only one track. Because, PhDs in this field or masters during this time can do so many different things. And one thing you have to identify is, what are those things? What are the different options available for you? So for that, you guys have to create a network of mentors and mentees. So find mentors or find people who are in a particular field that can guide you to go in a particular direction. But also find mentees, also find people who you can train, so that, because when you can talk, when you talk to these mentees, when you talk to these trainees, you learn a lot. I take my mentorship very seriously, because talking to my mentees always help me to see a different aspect, to learn a different thing. And while I'm helping my mentee to achieve what they want, I am learning a lot, and I am thinking in a different way as well. So from this slide, I want to tell you that, you don't have to think that, okay, if you're doing PhD, research is your only option. Think about what other things could inspire you, what you really love to do, and choose your career based upon that. And I think that takes me to my next slide, that choose a job that you like, and you never have to work another day in your life. So, I always thought that, if I'm passionate about my work and what I'm doing, I don't have those Monday blues on a Sunday night, right? So you want to look forward to your Monday morning when you go to work, that, okay, I am working for my passion. I'm going to change the patient's life. That's the drive for me. So work for your passions, and not just think, okay, what will give me a higher salary? What will give me more power in terms of managerial, more managerial power? Think about what moves me, what really drives me. And I know a few years back, I think almost 10 years back now, 11 years back now, I think I did a survey on a Nature. I think Nature has Nature, which is a journal, has a survey which can help you understand, based upon your strength and interest and your passion, what career path could benefit you. And I think I'm sure there are a lot of tools. I know available now on Google, because I was talking to my daughter and she was saying she took a test like that on a Google. So I know there are a lot of tools available online these days which can help you to determine, what is your passion, what are your strengths, and what career path can fit you. Another learning topic I want to tell you is, you are at the driving seat of your career. Think about, where you see yourself in 3 years, in 5 years, and in 10 years. You might have some idea about it, but it could change as well. But have some thinking in your mind, where you want to be in coming years. So, and start to accumulate those skills today. So for instance, when I became the lab head, when I was starting to run my lab, I did not know how to manage lab. I was managing the finances for my lab. I was managing 6 to 7 postdocs in my lab. I was being, the project leader, where I'm watching all the projects. I was the grant writer. I was writing my own grants. I was the manuscript writer. I was the only person writing my manuscript. I was the brainstorming person. I'm thinking about all these ideas. But I thought that during my career, I have not prepared myself for all those things. I was doing all those things. When you are in that situation, you will do it. But I think we can do those things better if we prepare ourselves. So right now you're doing your PhD and master's. Start to think about, if I want to run a lab, what skill sets I need, and start working on that. If you need to learn how to write a grant, how to write a manuscript, start working with your faculty. Ask them, can I help you to write a paper? Can I help you to do some research? Because those skill sets will help you prepare for your next step. So keep the future in mind and start to learn some of the skill sets. And also, like I said in the previous slide, developing your network is very important, because it helps you to understand what different pathways are available, and what is like, for instance, if I'm in a medical affair, what is my day-to-day look like? So understanding of that day-to-day life will help you to understand, do you want to do that work for rest of your life? Do you want to do that work for next 10 years? So that can help you to decide on which career path you want to choose. And here in this slide, I'm coming back to where we started from, that success is a journey, not a destination. Just keep learning and keep growing. Don't just think, okay, once I'm in this master's program, once I'm this PhD, I'm done. No, because that's what I learned from my journey, is that, when I thought I finished my PhD, I thought, oh, just getting this fellowship, my life is all set. It was not. As you go into the next step, it opens up many next steps for you. So I would say, enjoy this journey, enjoy this path, keep learning. Don't take it as a pressure. Don't take it as something that you are about to do, because if you're working with your passion, you will see that this is actually something that you love. This is your hobby. This is not some kind of pressure that your parents want you to do. This is something you want to do. You want to grow in your career, and this is something that makes you moving, and that you want to spend rest of your life or rest of your 10 years doing this particular thing. So keep that in mind. And I'm saying a lot of things because a lot of ideas are coming to me as I'm speaking to you guys. But I think I will end here saying thank you to all of you for your attention today. And I'm happy to take any questions.
Oh, thank you, Dr. Gandhi, for such a wonderful talk. You started your talk by a very pertinent topic about girl child being a burden, and I think your journey has just proved that what girls can do if they get some support from their family. And kudos to your parents for giving you that support, and see where you have reached. You have an amazing accomplishments as a researcher, And again, I think your passion, it's only your passion, because as when we are doing our PhDs, when we are doing our research, most of us have this single-minded goal to become an assistant professor. And you went to a foreign country and became an assistant professor there. And after achieving there, many of the people would say, okay, I have reached where I wanted to be in my life. And then you decided to take on another passion project of yours and move on to another journey. I think that is commendable, and that's just the something that I think everyone, including us, not just the students, that we should keep in mind, that we should always follow our passions, no matter what the society thinks, no matter what you initially wanted to be, because your passions and your goals also change as you change as a person. So hats off to you for such a wonderful journey, and thank you so much for sparing your morning on a Saturday and sharing your life experiences with us. And I also, personally like that you acknowledge your PhD guide. I think that's a very important thing, I think, which all of us should do, because you hear so many negative things about your supervisors from different people. So when you talked about that you're, so that, I think that is a positive sign. Everyone should know, your supervisors are also a reason for your success in life. So I think that was also important. Then emphasizing on the fact that, PhDs don't really have to follow just a single path. There can be multiple paths that they can take. I think that's also a very important point, because when we are, in our academics, we live in a bubble, and we always see scientists or assistant professors around us, and we forget, that there are other opportunities also available to us outside of academia. So I think that's a very important point that you highlighted. So yeah. I have just one scientific question before I, start taking the questions from students. You mentioned about the microRNAs. So I also work on non-coding RNAs, and you talked about detecting microRNAs in fecal matter. So I just wanted to know whether you were looking for microRNAs in exosomes, or how did you deal with the ribonucleases present there?
I know that's an important. First, thank you, Dr. Chabra, for summarizing my talk, and also I want to say one thing to all the teachers. We sometimes forget that how students are just looking at us. We are setting up examples for our students. What we are today, that we are, our students are going to be tomorrow. And I can vouch to that. What my mentors, how they have treated me, this is what I'm taking to my in my journey and teaching it to my mentees. So that, just keep that in mind, because we think that, oh, students don't care about us, but students are watching you and they are going to follow you. So you are an important component of their life. About the microRNA, so I was not just testing the exosomes, because exosome biology, when I started to publish, I noticed, there were several papers that were looking for exosomes, but I found that actually these microRNAs can be present even without exosomes. So that's why, and I wanted to look at the complete signature, not just the exosome signature. And when I started to work, I think exosome techniques, purifying exosome techniques were not very clear. But when I did when I got the funding from NIH for biomarker development, I worked with a colleague whose specialty was actually in the exosome development. And I know he made some strides there. So yes, short answer to your question was complete microRNA, because exosome technology was not very charted out 10, 11 years back when I started to work on.
So what about the ribonucleases? Did you face any difficulty with that?
I know in stool, there are a lot of ribonucleases. That's what we were saying. We were saying, we said that, this is finding a needle in a haystack, right? We have so much microbiota, so much microRNA in the stool samples. And we were thinking, if everything survives all those enzymatic attack and other things, then I think we have the right signature. And I know one of my colleagues is now continuing with that study. I have to touch base with him again, if he's, what's the next step for him.
Okay, thank you, Dr. Gandhi. And before I hand over the stage to Dr. Manju Jain, I think it's also important to thank her for starting this very important series. I think it's a wonderful initiative that she has taken, and I think all the students from our department will get a lot of benefit out of that. So Dr. Jan, do you want to take the questions?
Yeah. Thank you, Dr. Ravindresh, for the vote of thanks. And yeah, I really wanted you two people to interact because he is a new joinee in our department and working in the area of long non-coding RNAs in context of cancer. So Professor Ramakrishna, if you have any questions, please. Sir, you have to unmute yourself.
I don't have any question, but I saw the slides. They are quite very simple way she has put it, which is good, because immunology is not an easy subject to understand. And the papers published in Nature Immunology or some of the top journals, she explained it very nicely. And also I saw initially the issue in India with the girls. But I don't think now it is like that. Yeah, that may be an old one. I think a lot of revolutionary changes have taken place. Maybe it is still there in some backward areas or villages, I don't know, but at least in where we are living, we don't see too much of that. Although there may be some inherent kind of this thing going on, but we try to negate those or try to give women all equal rights and positions. And yeah, what you said, going from a student. So I have gone through all that path that you went, maybe most of it. I was in the US for 18 years. So I did my postdoc, I joined their assistant professor, associate professor. Then I came back here in 2015. I came here because I really wanted to come here because I spent a lot of time. Okay, I could have spent there my rest of my life and then I would have retired there. But I thought that I should get something back here in terms of teaching, research, back to India from where I am coming from. So I really liked interacting with students, teaching them, and whatever you are showing in the last few slides, that I keep telling them, at least the second year students who are there, they probably will tell the same thing, that I tell them, do what you like, what you love to do in your life. Don't do because somebody is pressurizing you. So whatever it is, whether it is science or something, not even science, you have to enjoy what you are doing every day. When you go, the same thing that you are telling, that it should not be like work, it should be more like play, right? Yes. You never get tired if you are really doing what you love to do at work. So those are the similar things which are coming out. So I can see a number of second year students here, although I have not interacted much with the first year students. So yes, overall, I think it was a great seminar, and it was, I hope all the students learned, and let us see what questions they have. Thank you very much.
Yeah, thank you, sir. So now I would ask the students to take up their part, the whole purpose of the entire show. And from the fourth semester students, it's Chenika and Pranay who will put up questions on behalf of the students, and I'm really happy some of our pass out students I can see there. So Piyush, it is nice to see you. Others I'm not able to recognize. Yeah. So yeah, Chenika, you can take up some questions on behalf of the students in the chat box. You can see.
Yes, ma'am. Good evening, ma'am. First of all, I would like to thank Rupali ma'am for this wonderful talk. It was very informative for us, and I'm sure all the audience must have learned a lot from you and your journey. So I will take some questions from the chat box. So the first question is by Lavlesh Thakur. She's saying it was a wonderful, informative lecture. My query is, how would you relate the changes in gut microbiota of MS patients with the cure or treatment of the disease? I mean, how immune response can be related with changes in gut microbiota?
Yeah, so gut microbiota is a very important feature of the immune system. So lot of immune cells, actually lot of regulatory cells are actually in the gut, so there is a lot of immune system activation, and that's why remember those days when you eat something and you don't feel well. So that's what happens, when you eat something, your immune system gets very activated, but there are a lot of regulatory cells that calms the immune system down. So there are a lot of publication that actually shows how gut microbiome is related to immune system, and then also how gut microbiome is related to neurologic, how you think. So there are a lot of what do you say, lymph nodes present in close to our intestine that actually are modulated by the gut microbiome present. And there are studies, that have shown that once you take the probiotics or the good bacteria, how it can help to calm your inflammation or immune system and help you with gaining the health back, help you getting the health back and overcome the disease.
Thank you, ma'am. I'll take the next question.
So Chenika, I
So
think
it
you
is
can,
by Abhijit Nayak.
excuse me, Chenika, you can go ahead with your questions first. I can see in the chat box, right?
Yes. Yes, ma'am.
Yeah.
Yes, ma'am. Ma'am, my question is, so what kind of research approach we can take further for the aryl hydrocarbon receptor work to like induce T cells? What can be done by us later on? So,
Yeah, so I think if these HR regulatory cells can help the patient. So one thing is, maybe we can give some of these ligands, and we have to find the ligand I work on was the TCDD, which is a toxic ligand. We cannot work on that, but I worked on that particular ligand because that is a very strong activator of AHR pathway. But I know there are some non-toxic ligands, and I published in collaboration with other colleagues, where we used ITE, which is another ligand for HR, and that is a non-toxic ligand, and they have used that particular ligand in the colitis model. So in the next step, if we can drive or make a molecule that can activate the HR in the body. But when you're giving something to the human, you have to go through a lot of testing and making sure that it's a safe for human clinical trial. So maybe going towards that direction, making a molecule that can really help the patient with the disease, and also making sure that the molecule that you generate is safe. So I think, and beside that, right now we tested this AHR on T cells, but also on dendritic cells. And I'm sure because HR is present on different cell population, we should understand how this HR activation on other cells affects, because, right now we are seeing one side of the story. You want to understand the whole story before, you take it to the clinical trial or helping patients treat the disease.
thank you, ma'am. Thank you. It was really very interesting to look forward to. So now I'll take the next question. It is by Abhishek Kumar Maurya. He's asking, what are the new techniques or skills one researcher should acquire or learn to develop biomarkers for any disease?
I'll start with the first thing you should learn is patience. Because patients, biomarker research, when I joined, I became the head of the biomarker team and was managing the biorepository. I was thinking, there are these 2,000 patients. We have like thousands and thousands of blood samples from them. I was working with, top-notch biomarker experts. I'm like, what's the problem? Why can't, I'm like surprised why nobody could find a biomarker for MS. And I was like, so positive we will find it. But, the biomarker research is time taking, need lot of resources in terms of patient samples and lot of funding, because all these assays are not easy. So the skill sets you guys can learn right now is understanding various biomarker techniques. What are different biomarker techniques? And right now there is a lot going on in terms of omics. There is proteomics, there is micrometabolomics, there is this gene omics, gene sequencing studies. So lot of profiling assays are going on, and I think starting from there is a good thing too. So understanding what assays are available and understanding the analysis method for those assays, because if you're looking at 100,000 things or 500,000 genes or microbiomes or metabolomic outcomes, you need to have techniques to analyze them. And so that's why I was working with a biostatistician or bioinformatician who were helping me to finding the needle in the haystack, because when you have such a diverse array of things that you find, you have to first find out what of them is meaningful and what of them is actually some significant finding, instead of some just noise. Because you tested 500,000 things, you will find 50 things that are actually different, but you have to make sure that those 50 things are correct. So I would say, develop a lot of patience, learn about these various omics method, and also learn about various analysis method that can help you take you to the biomarker path.
Yes, so I think another question from Chenika, which is a very naive and I think useful question, which she is missing on. What is the work that you do as a medical director? Tell us.
Yeah. So I try to touch it on a little bit. So as a medical director, I think my primary job is to learn about all the data about the products I work on. So I, like the treatments that I work on, I know what happened in their clinical trials, what is their label, their prescription label looks like, what are the safety concerns with this treatment. So I am like kind of the knowledge person, because everybody who has question about my treatment is going to come to me to know what research has been done, what clinical trials were done. So I need to know all that data. And so one of my primary job is to answer questions related to the treatment. The second part of my job is training people. So I train people who are in commercial, in marketing, in medical, about this data, about immunology, about the disease. So that is the second part of my job. The third part is, I work on various strategy committee where we think about, okay, what is missing? What is the unmet need right now in the field? What are the questions that neurologists are asking us, and how to develop material for that? What are those material? Then I also review a lot of material that goes out in medical field or commercial team. There are a lot of resources that are sent out to doctors, right? A lot of resources that are sent out to patients, and I have to make sure that all the information, all the medical information on those resources are correct. So those are some part of my job. I hope I explained it well.
Yeah. So next I would maybe ask Priyanka Madan, because she's her PhD project is in and around from psoriasis, another T cell based autoimmune disease. So she is working in that direction. So Priyanka, you have any question?
Ma'am, one thing is, it's just a very basic question. So you went for the miRNA seq on Mira seq for these plasma, serum, or even for the fecal matter?
So no, at that time, mRNA, microRNA seq were not available. And what was the name of Exicon? So they had an array of 400 or 800 microRNAs. Exicon at that time had an 800 microRNA kit that I used. I know now Pacman has that kit too, but that was not working for me. So this Exicon kit was more specific for serum and plasma, and they have assays at that time. And I'm sure there must be a lot more right now, but at that time, the Exicon assays were giving me a good quality control in terms of microRNA analysis in serum and plasma.
Okay, ma'am. Ma'am, one thing, I just saw one of your slides where you showed a few set of microRNAs with respect to the relapse of MS and different progressive stages. So with the study you did, there was set of microRNAs, or you were sure of that these could be used as a biomarker for MS or different stages of MS? You found out,
So,
or
Sorry. Yeah. So that was actually the analysis. I did that. Tested 800 microRNAs, and I showed that funnel thing, right? So initially, I did a study on, I think, 20 patients. Then I did a study on 50 patients, and then I did a study on 150, and then 400 patients. So I took these 800 microRNA, tested them in different groups of patients, and the slide that you were seeing is, I think, analysis on about 100 patient, where you could see that how these microRNA is changing in different group of patients. So not what should I say, hypothesis driven. I did not know that these microRNAs will be present, but I started from scratch and build it from there. And then when I wrote my papers, I found that some of those microRNAs were overlapping with other publications that studied microRNAs in MS versus controls, and I discussed them in my papers.
Thank you, ma'am. Thank you.
Sure.
So Priyanka Madan is asking this because I'm asking her to do a rigorous, search for these microRNAs in context of her disease. So she's like, okay, what is some, sort of streamlined path? So definitely
Yeah.
you have to really, put yourself inside and come back with maybe one or two important molecules.
Yeah. So yeah. Or
So
you
it
can do
has
a
to,
search and start from there, depending upon resources, right? If you have lot of funding, then maybe start from this omic thing. If you don't have lot of funding, maybe start from what is already published and test those microRNAs.
yeah. So that way things are going in parallel. So first I put her into literature. So as I get more money, I'm getting into the RNA-seq stuff. Yeah.
Awesome.
So
Thank you.
yeah, in context of training a PhD and from a PhD student perspective, I would say, is it that like I would like to touch? I see you talking of multiple projects being taken up in a single go. Definitely not in a randomized way. There is some relation, right? Because one thing definitely gives rise to many branches, and finally it does make some sense, the holistic picture. So generally, like as a PI, so I'm discussing, the tussles of a PI with another already, a lady with so much of experience. So generally, definitely we got to have plan A, plan B, and plan C. But somehow I would like you to give this message to the students. So we really have to together, pull in the resources available to really make something out of what is available in terms of the literature, and to take our project in a original and novel way. For that we have to explore more than one window.
Yeah, of
And
course.
that,
Yeah. And I was talking about 44 publication published, but I'm telling you, I worked on multiple projects, and I was actually crying on the day when I was cleaning up my office, going to industry. That I was, so many of those folders were filled with negative data. Because you will start with so many projects, and out of them only a few of them are successful or made to the manuscript. So first of all, do your search in terms of finding what is an unmet need. Don't go for everything that everybody else is doing. Go for things that you think will make answer a big unmet need in the science, right? What are those questions? What is really making you tickle, not letting you sleep at night? Why this particular molecule is interacting with this molecule and generating a regulatory T cells. So think about, what is keeping you at night, what is bothering you, what is an unmet need in terms of the biology field, and think about the project from that way. And even when you do these projects, take it like, okay, you're learning something from these projects, and all these projects won't give you a publication. Keep that in mind, but they will always teach you something.
so
I
that's a
really,
difficult, yeah. Please go
I
ahead.
really want to promote publication of negative data, because there was so much negative data. I would have like 500 publications.
So I really appreciate your take on that. I would be in for that. Yeah. So yeah, I hope the students have taken the right perspective, and definitely she has given up a little hard condition. Assume that you will not have any publication out of it. So really, I'm telling you, don't worry about that. As long as you get hold of the right problem and possibly the right solution. As
Yeah.
peripherals, you will get something out of it. At least you
Yeah.
will be trained well, right? How to
Yeah.
approach
Yeah.
a problem.
That's right.
And in the
Yeah.
long term, that becomes very important. So I would again give the things back to student Pranay. Yes, please go ahead with the questions of your batch, maybe, because I want the junior batch also to come up with things. So take up the questions from your batch. Or, yeah.
Yes, ma'am. Yeah,
Yes,
thank you for the
Asim.
opportunity. Um, there was a question from my batch. Tanya Bansal has asked, can we predict autoimmune diseases occurrence in a person?
Oh, can you repeat, Pranay? Your
Hello.
sound was not very clear to me. Can you please repeat it?
So maybe I can repeat the question for him. This is from
Hello,
Tanya, right?
ma'am. Can you hear me?
Yes, Asim, be a bit louder.
Yeah, sure. Tanya Bansal has asked a question. Can we predict autoimmune diseases occurrence in a person?
Oh, predict. Oh, so there is actually,
Yes, ma'am.
right now a lot of
Can you hear me now?
genetic study. Yes, I can hear you, Pranay. I got your question.
Hello.
So right now, there has been done a lot of genetic analysis on these autoimmune patients, and there are some history. Sometimes these some kind of genetic association can cause autoimmune diseases, and specifically for multiple sclerosis, I know there is a gene signature. Manju, I'm resonating again.
So
I
maybe I can.
Yeah. So sometimes I know that in multiple sclerosis, there are multiple genes that have been linked with the disease. So yes, if there is a parent who has the disease like multiple sclerosis, their kids, if they have the same gene, can have the disease. So, but one part of that job right now, what I was doing as a medical director was looking into this genetic screening and genetic counseling. So as you know that there are certain ethnic group who are more prone to certain disease, and these ethnic groups actually have genetic screening available to them where they can test what genetic makeup is there for them, and then decide if they want to have a kid or not, because they could carry some of the genetic mutation that they have and can carry the diseases they have. So yes, you can predict them, but I think it's not very widely accepted right now, because it is a very sensitive matter, and I think the medical field and people have to really understand these genetics and their association with disease in detail, before we really make it very popular in terms of our understanding. Does it answer your question, Pranay?
Yes, ma'am. Ma'am, I have a question from my side. What mouse model
Yeah.
did you use for the MS?
So I did not work on the mouse model, the publications I showed you, but I know there was a EAE model, the experimental autoimmune encephalitis model, that is available for MS, and I think that is used by immunization with MOG and MBP. And I know there was another model being used, which was on nod mouse, that is more similar to progressive disease, is used. But there are tons of model available for MS study, depending upon what cell types you want to study, because I know there are some studies on the blood-brain barrier, and for that there is a totally different model than these two models that I talked about.
Yes, Pranay, maybe we can give the stage to now the first years. Yeah. So Abhijit,
Yes, ma'am.
you would
Thank
like
you,
to
ma'am.
come up? Yeah, thank you, Darshini. Abhijit, you would like to come up with last 2 to 3 questions, maybe? I don't see Chitransi here.
Yeah.
Yes,
Ma'am,
Abhijit.
yes, ma'am. We have
Oh,
ma'am,
you are there. Yeah.
we have questions from our batch. Uh, Ma'am, how is applying for a academic career different from applying for a industry job? That is, which skills are focused more in academic or in industry? Like different.
Skills. I think so. Depending upon what job you want, right? I think one skill set that really helps in academic or industry position is people skill, right? Um, Listening skill, so there are some soft skills in terms of how you interact and communicate to the other people. So some of those skills are really helpful. Like for instance, when you are giving a presentation, so I'm talking about the skills that can be used for either one of them, and then hopefully I'll be able to point out what skill could be used in one versus the other. So if you're looking at the common skills that help is the communication. So when you are giving a presentation, you're giving a clear, concise presentation. When you're answering questions, you are able to provide a precise answer. So those communication skill will help you in either job. The second skill, I think one skill that can help you in industry and having a business aspect, having a business perspective in mind. What is the bigger picture? What is company wants from this particular project, and keeping that in mind could help you. For research in academia, I think it depends upon the PI. If you're approaching a PI with certain interest, like for instance, if I'm a biomarker person and if somebody is looking for a postdoc with me, I will need a person who has experience in this omics techniques or experience in analyzing a big data. So those skills will be helpful. So depend in academia, it varies from which project you worry, but in industry, having a business, keeping that understanding of business in mind can help you.
Yes, ma'am. Another one question is there. Uh, Ma'am, how studies done on mouse models are ensured that it will show similar effects in human body also? That is how work on mouse is correlated with humans.
That's $1 million question, Abhijit. And that is my biggest concern right now. A lot of mouse models are not humans. So many, you will see tons and tons of research of different molecules working in the mouse model, but then when they were tested with human, they are not working. So mouse are very different than human, although a lot of treatments for multiple sclerosis came from mouse studies, but there are a lot of studies that actually failed as well. So mouse model can give us some hint in terms of what could work in human, but I think it does not provide us always the answer.
So yes, Abhijit,
Yes,
maybe,
ma'am. Thank you.
yeah, thank you. So I would like to portray one question from Hassan. Again, a very naive question that almost every master's or maybe graduate student must be wondering, having maybe the right or the pseudo effects. I don't know, you can clarify for him. Since you settled abroad, madam, you have observed many scientists and researchers with many specific characteristics that you notice in them, which we can build into. So portraying it, are we lacking in something, the Indians?
So it's not a naive question. No question is naive. I think because where you are, you want to learn more. It always, that's a question in your mind. So I don't think Indians are lacking in one thing. I think one thing that we can learn is the flexibility. Sometimes we are very stubborn in terms of the idea we have, or the project we have, or the opinion we have. So I think one thing that can help us in early stage is having that flexibility in mind, in terms of career choices and in terms of understanding. So there is so much to learn from others. That's what I see. Every person I talk to, I learn something from them. There is so much to learn from others, and keep your mind open for that learning. And I think that is one thing we can learn. And I think Indians are not lacking anywhere behind. We are hard workers. We work very hard. We want to do good. And I think that characteristic we should always keep with us.
So I want to add something there. To Dr. Rupali, because I have been there for a long time. So I'll tell you, a lot of students there come from India and China, okay, predominantly. Many other countries. So if you compare that, Chinese are very, very hardworking. They will sit in the whole night and they will work there. But the advantage we Indians have is our English, okay. We can speak English a lot better. People there can understand than Chinese who is coming newly. But Chinese also, if they're coming at a young age, they really do very well with their English. They will pick up. So that is one good thing. As she said, that we need to learn from the American system is the way they think. They open mind. Okay, there is no closeness in their thinking. Okay, they are very open. If you look at American professors and all, there is no, okay, this person is coming from this country, that kind of thing is not there. And there is no judgment. They don't have fixed kind of idea. That is, I think, very important in science to have that openness and think in different directions. Don't think, I have this set idea, this is it. It can happen 10 other ways. So that is important to think.
Yeah.
Okay.
Can I add one thing that's well said, Dr. Ramakrishna? And one thing that I was lacking in my career is, keeping the bigger picture in mind. When I was in postdoc, my only interest was work on this human experiment and this, keep the bigger picture. What I want to really change the world in a bigger way, right? So keep that bigger picture in mind. Don't just get too much nitty gritty in the experiments that you're doing or the project you're doing. Keep thinking about, okay, what is the unmet need, and ask questions. Never hesitate to ask questions. Never say, oh, is this a dumb question or is this a right question to ask? You have the question, it means there is a problem or there is an understanding needed. So don't hesitate to ask question, and don't hesitate to question something. If you see a manuscript well written in a Nature Immunology, don't say, okay, I just accept whatever is written in the manuscript. Ask, did they choose the right mouse model? Did they find the right ligand? Was their study correct? Even if it's published in Nature Immunology, it can still have problems, right? So ask those bigger questions. Never hesitate for that. And you could just learn from by asking questions. So keep those things in mind.
Yeah, I have one question that I would like to ask, which I kind of get very often, which is there are students who are not interested in doing bench work, but at the same time, they don't want to leave science altogether. So what would your recommendation be to, let's say, a master student who wants to stay in science, but doesn't want to do bench work? Would you recommend a PhD to such students?
No. If you cannot do bench work because your majority, so PhD, so there are now fields in PhD. Sorry, I was not thinking of that direction. So PhD in certain areas won't need bench work. Like for instance, if you do PhD in bioinformatics, right, you might not need that much of a bench work. If you do PhD in a field which involve data analysis or biostat, maybe you don't need that much bench work, and more in terms of setting up formulas or learning some of the computer algorithms. So if you are not interested in bench work, please don't do PhD in a project that involves a lot of bench work, because it will only bring frustration to you. So think about what other ideas you can pursue and do a PhD with less bench work, or think about a career. Everybody doesn't have to do a PhD, right? You can still set up a successful career path even without PhD. And I'm sure even at master's level, there could be some trainings, like for instance in medical writing. We had a speaker last week from the medical writing. There could be some of the trainings that you can do right now instead of PhD, that fields that really excites you. So maybe listen to your passion, what you really want to do, and then do those things as your next step instead of pushing yourself just because normal PhD should do this bench work. Don't go in that direction. Please listen to your passion and what you really want to do.
So with this, maybe I can ask Chitransi, because she was nominated as the volunteer for today. So it's something she should be asking. Chitransi, your question or somebody else's question you can put up.
Hello. Thank you, Rupali ma'am, for this beautiful session. And I would like to ask a question of Nilottam from our batchmate. He's asking that what is the difference in approach of research between a research institute and a vaccine making or pharma company?
I think that's a good question. So, when you're in a pharma company, your research is very focused. Your focus is like if you are finding a treatment for lupus, let's say, I'm just giving an example. You're working on a molecule that is provided to you, and you will do research to make sure that whether this particular molecule can go further in the clinical trial or not, right? So it's more focused. Whereas in academic research is I would say it's like an artist, right? It's you have given a canvas and a paint, and you can choose whatever you want to, although it is a little bit restricted on what your PI is working on, what kind of funding you have, because you have to provide milestones to NIH and the funding agencies. But I feel like it's more like a open canvas, and you can draw in whatever direction the project goes. But in industry, I think it's more in terms of what is business, because business is developing this particular treatment and is more focused. But I won't say that that's always true, because in industry, like for instance, right now in my position, there are a lot of fundings to understand the disease state. A lot of work that Sanofi does is understanding the disease, what it is. So it's not always true, but I think still it's more focused than the academic research.
So with this, I guess if you can, all of you, I'm requesting maybe another 5 minutes or so. I would like, just on a personal note, I will ask Rupali. So do you miss the Gandhi lab, or it's perfect?
I do miss my lab, because your lab becomes like your family. I felt like it's a family for me. Those are all like very close, because you 10 hours you're working with those people. But one thing is, I am in touch with most of my people who were in the lab. So once in a while we meet in the meeting, because we are still pursuing the career in the similar direction. So we meet in the conferences, which is nice. I hear about they getting married, they are changing jobs, so I learn about that, so which is good. But I, at some level, I do miss that, going with the open mind in the lab and thinking, okay, what's coming for me today? What is the great idea coming, right? But here at Sanofi, the passion for science and the project is so much. I didn't miss the Gandhi lab. Some aspect of it I missed, but not working in the lab, or like not able to contribute to the patient. So that part I could carry with this particular position.
Yes, and I really remember like the kind of student Rupali was at her time. I mean, her PI had to literally drag her out of the lab. Okay, it's time to leave. It's enough. So this was the way she was working, right?
Yeah.
It's all passion that.
At that time, the only passion I had is I have to get the PhD. Very focused mind in terms of setting up this thesis and finding something from out of my PhD.
So she really, I think she owned her work to the highest extent one can. So that is really appreciable, and I think one should be inculcating the passion component of the entire process. It's very important. It drives you in the the right kind of training, basically. So with this, just one question from my side. you talked of, and you have spent a lot of years working on markers for different neuroimmune diseases, I would say, like the MS, SLEs, and many more. Uh, so I think we are stuck up with the point with respect to the early disease markers. So how good we are, I mean, at present context, because I'm definitely not updated. And other thing which is like conjoined to this question is I myself maybe 4 to 5 years back conceptualized a project wherein if I take all these neuroimmune kind of diseases and try to review them from an immunological perspective, actually. So I say I'm trying to do immunology. I'm not saying I'm an immunologist, right? I'm on the path to establishing myself on that track. So I see a lot of similarities in context of the immune neuronal accesses that are being, altered in not one but multiple neuroimmune diseases. And the kind of question which I wanted to address, because I'm not getting a neurobiology based collaborator, so I do plan to execute that project in the upcoming times. So my question was, okay, a lot of peripheral immune changes happen, and then they translate into the brain immune changes, the neuro immune changes, and then it leads to possibly the neuroinflammation and then neurodegeneration and the disease specific output in terms of the neuronal behaviors and parameters. Or it is like other way around. Other school of thought is it comes, the origin is from the brain itself. Something goes wrong there and then it travels to periphery. So what is your take on that? Because I tend to have the first thing, I don't know how much correct, I mean, based on literature, both kind of schools exist.
Yeah, so I wish you good luck, in your collaboration for understanding the neuroimmunological axis. So there are right now two school of thoughts, because I worked in immunology, my understanding, and I think once you work on certain line, you start thinking the same way. So for
Exactly.
20 years, I studied immune system, and I see so many things altered in the immune system for these patients. So my understanding is things start in the periphery and goes in the brain. And I give a lot of talks about that, during my current career. But in some part there are things wrong in the CNS as well. So it's not one way or the other. I think it's combination of things that leads to these progression of neurologic diseases. So because we can change something in the periphery, I think that gives us some tools. Like all the treatments right now for multiple sclerosis are targeting the immune cells in the periphery, right? And we could find not a treatment for MS, but at least something that helps patients or delay their progression, right? So seems like that maybe that hypothesis is correct, that things go wrong in the immune system in the periphery and goes in the brain. But for you, your other comment about being commonality among various autoimmune neurologic diseases is actually true, because the regulatory T cells, the role of regulatory T cells, the role of some of the co-stimulatory molecule, the role of inflammatory cytokines has been shown in multiple disease. And that really surprises me, that although maybe at a bigger picture the similar alterations are happening, but why some people are getting arthritis, other are getting lupus, and other are getting multiple sclerosis. So if we find an answer for that, maybe we'll cure all these diseases.
Exactly. So that was my take on that, Rupali, because definitely I'm not a specialist in the area, but my thing, I could find some common immune accesses getting altered, although in different directions. So somehow if we can get hold of those accesses and try to sort of kite fly with them, so it will give us answers to not one but multiple diseases.
Multiple diseases. And for sure, that is true, Manju, and maybe you're correct, and that's what people have done, right? But what happening is with these 20 multiple sclerosis treatment, it's not working for all the patients, right? There are 30-40% of the patients who are not responding to this treatment. So maybe in that particular patient, there is a different situation, there is a different equation going on, right? So I think with the help of biomarker, if we can identify, put these patients in different axis in different buckets, what axis is going wrong in these patients, and targeting that particular pathway could help us. But I think it's too much to ask. When you will look at these patients, it's, oh my God, they are so heterogeneous. There is so much variability. It's really hard to put them in these buckets.
Because whatever I'm coming up, it's just because, as I told you, I haven't really started in the field, but it's just out of the literature, the idea I'm getting. And is there any study that tells you about the time dependency? What has happened first, and then it followed, and then it followed? I really wanted to, sort of
If we
do
know
a
this
time
will
dependence.
find cure, right? If we know this will find cure.
Exactly. So that was the model I had proposed in that project.
Yeah,
It's still
yeah,
lying in, under my files, because
yeah.
then I have to go slow.
Yeah, good luck. Yeah, I think that model will be very helpful if we know what goes wrong first, what comes second. Although we have some ideas, right? We have hypotheses, but does it the same pathway happen in all the patients? We have to confirm that. And if it happens only in 10% of the patients, maybe we have to identify that particular pool and work with that pool.
So, Dr. Rupali, there is a question from a student, Gargi. She's asking that since you said PhD advisor is very important and they contribute in all our careers and all. So, how do you choose a PhD advisor? How do you know this person is right? So, please answer that.
Oh, I know. I know it's very, very hard, right? I would say, for me, I was lucky, or universe has provided me my mentor, and I know Manju, and a lot of people know him, and we all know how awesome he is. So we don't get I do not know how much choice you get while you're choosing the PhD advisor, but I would say talk to the students in their lab, and see how this advisor is this advisor committed to students development. Is this advisor help you go to your next step? Is this advisor, how do they do with the challenges, because all projects are not going to work. So is advisor get scream at students like you are not doing the right job, or is he helping you to find the answer? If there is a problem, if there is a challenge, what is the solution for that, right? So I think as an advisor, those are our responsibilities. Instead of blaming the students that they are not doing the right job, and students have to make sure that they do the right job, they are doing their essays properly. It's not that they are running a Western block and they go for a tea and the Western blot goes over. So do your essays properly as well. But from mentor side, they have to support the student. So the only way I think you can find out the right PhD mentor is by talking to the students, their students, which I have not done because, I was just lucky to find a good mentor.
So it is very important to talk to the people, as Dr. Rupali said, because sometimes what happens is students just look at publications. Okay, this person has papers in Nature, Science, so it is great. But sometimes publications can be very give you the wrong information, because they may have great papers, but in the lab, I know some people who are very, very mean to the students, okay, and their careers have been spoiled, and people have to leave the US and come back here. So that's an important thing. As she said, talk to the people, and sometimes you may not get straight answers by talking to the people in the same lab. Maybe other people around the lab, in the institute. Okay, all those things will help.
Yeah, networking is one thing, and I know I have a lot of hesitations in networking, and I still struggle with it. But I think networking is important. Is just talking to people and learning from them.
Yes, so with this, I think we have really, we are at the end of the session with very constructive discussions, useful for the students, I believe. And I would really congratulate Rupali for being so, open for such things. Not now, I've seen her like right from the beginning. a small thing I would say, we would travel back home. Many a times we were accompanied because of one situation or the other. In a bus, there's a bus event. So while we are like stuck up in that, DTC crowded bus, she is there and I'm at the back, and we are talking. I'm trying to, share some concern. She's like, "Bolte raho, I'm listening to you. Keep talking." So that is the thing, the kind of interaction, the openness. So it really, make a whole lot of difference. You got to talk if you have anything going at your background, so that it can be resolved.
Yeah,
You got
yeah,
to connect.
yeah. Don't keep your problems. I'm sure you will. You have to approach 10 people, and maybe 2 out of them will reply to you. But keep making your efforts. You will find, what do you say, inspirations and answers from people you will talk to.
Yes. So on behalf of the Department of Biochemistry and our university, I really thank Rupali for being there so early in the morning. And with this, I will conclude today's session. Before that, I would just like to flash the next speaker in the series. I think he is with us. So that way, there is this connection, one connection to other going on. So you can see my slide.
No, it's not visible, ma'am. Your Google Meet is flashing. It's not your slide.
Wait. Actually, because I hooked him there, no? So I think I should be sharing it. Hello, Dr. Narendra. He's there with us. The next speaker, the third speaker of the talk.
Hello, Rupali.
Hi, Narendra.
It's very nice talk.
Oh, thank you.
Here, my son is asking something that is. Wait for 1 second.
No problem. Now you can see the slide, Dr. Ravindresh?
No, we cannot see your
no,
slide.
ma'am, it's not. No, we cannot see it. Yes. There seems to be some trouble. Right now, you're not even presenting your screen. You need to, present now and go to the slide and share your entire screen.
Somehow that option is, not. Anyways, we have Dr. Narendra. So he's another associate of ours. He did his PhD from JNU, and that is how we know each other. And right now, like, he went for his postdoc at Uppsala University. Yeah, it's in progress, I think. Yeah, now it is showing up.
Yeah, we can still see your Google Meet thing only. You just go
because
to. Yeah, now it's
okay?
visible. Yeah.
Yes. So Dr. Narendra Pradhan will be the third speaker in the subsequent, lecture, and I do have to confirm the dates and the time with him. And it's interesting that with a whole, years of input in research, he describes himself at present as a researcher and an entrepreneur. His area of expertise is cancer and vascular biology, and right now he is associated with the specific business startup programs at Uppsala Innovation Center, and actually is a founder and director of Uppsala-based startups. One of them named as Molecular Kitchen, and I believe he is in the transition phase, and that becomes all the more important for all of us to connect with him, because this is the need of the time. So just some examples in front of the students, so that they have a lot of hope in the years to come, and that hope makes them work with more enthusiasm and happiness. That is the whole take-home message of this entire series. Yes, thank you so much, each one of you, to join us. Yes, thank you.
Thank you, Manju, for organizing this.
Yeah, thank you. See you later. Yeah, bye.
Thank
you, Naren, for joining. Thank you, Dr. Ravindresh, for being there. I know you are on a weekend at Chandigarh, but sorry about it.
No, it's fine. It was an interesting talk. I'm glad to be here.
Yeah.
Thank you, Dr. Gandhi. Thank you.
Thank you. Bye. Nice to meet you all. Bye.
Bye.
Good luck. Good luck, everybody. Do good in career. Bye.
Thank you, ma'am.